Yeah, in this class, we will discuss about the plant tissue culture laboratory. You know that plant tissue culture laboratory is an important laboratory of a plant science. Now, in this slide, we can see need of low cost lab because we don't want to much more invest the in lab, so we are going to construct a low cost lab for the plant tissue culture. Even though if you are going to construct a low cost laboratory for plant tissue culture, we should not compromise in the quality of the propagation of plants, and micropropagation techniques is too much of costly. So we have to produce the micropropagation with uncompromisable cost. The potential increasing the interest in agriculture crop protection also increased. The low cost plant tissue culture technology will stay high priority in agriculture, horticulture, forestry, and floriculture of many developing countries for protection of the suitable priced high quality plant materials. On next slide, we are going to see about the general requirements of a setup a low cost laboratory. First, this is a layout of an identical or typical plant tissue culture lab with the separated washing room, dissective room, culture room, and working area also we have provided. And the location of the plant tissue culture is an important part because the transport and every services should be accessible to the lab, and the site should be very trained to the locally and other peoples also. And structural requirements, the structural requirement of the plant tissue culture, the root structure should be the roof reserve. The roof has to be timber or steel with anti rust paint and the ceiling must be at least 2.4 meter from the base or the finished floor lines, and the wall should be concreted smooth finished with anti-fungal apacitic that paints. And also the window shall be properly installed with 16 mesh screens at 1 meter of diameter. The main doors ways should be a 1.5 meter wide and moisture proof door should be provided. The floor shall be a concreted, smooth, non-skid, slope 2% to 4% towards the tray. And another, we are going to see about the functional requirements of the plant tissue culture laboratory. The basic facilities of the laboratory is concerned one area, room with a computer, with a locker for the keeping. Apron is the high pressure fan to clean the exhaust external materials. The working place or the experiment preparation and washing room, sterilization room, media preparation room, incubation room, incubation chamber also we can consider, then inoculation chamber, and very important is we need the skilled person for this plant tissue culture laboratory. The washing room, the glassware washing shall be located near to the sterilization as well as some media preparation rooms. The washing zinc should be 45 into 60 centimeter with the acidic as well as alkaline proof material. The room also provided with the buckets, detergents, mugs also, and everything should be provided. The room provides the water lines as well as the electricity lines. And the sterilization room, the room should have a autoclaved. If you don't have an autoclave, means the cooker is enough with a proper ventilation with exhausted fan. And also the sterilization room should provided the media bottles and autoclaves or cooker sterilizations in that room itself. The bottle should be sterilized 121 degrees Celsius at 15 lbs for 20 minutes to 1 hour. It contains a wrapping paper, wrap the rubber tight, inoculating the dishes for the sterilization. In the media preparation room, that room should provided with the working tables, the measurement about 1.2 meter of height, and it should be 850 millimeter to 900 millimeter in a wide range. The tabletop should be covered with a material which is easily cleaned with any waters or in disinfectant solutions also. The water sources and glassware storage here should be provided. Room contains double distilled unit, pH meter, buffer solutions, electronic balance, cooling freezer, measuring cylinders, magnetic stirrers, pipettes, hot plates, and also the media bottles also we should be provided in the media preparation room. And another one is we should have some stock solution reagents bottles to maintain for the stock preparation, and the chemical should be sucrose, adenine sulfate, and inositol, agar, and other vitamins or hormones also provided in should be maintained in the tissue culture media preparation room. And inoculation chamber or inoculation room, if your inoculation chamber should be in a aseptic condition, in the floor rooms covered with the tiles and facilitate the proper cleaning. The room contains the laminar airflow chamber, cabinet or the chamber with HEPA filters also. It requires the sodium hypochlorite, mercury chloride, and 70% ethanol for the surface sterilization. It requires the forceps, scalpel, sterile bottles, sterile dishes, split lamps, cottons, and hand cans also which has to be provided in this inoculation chamber. Then incubation chamber or incubation room or the culture room, the both are the same. Wide range of the we are going to culture that in that room itself, so we have to maintain a aseptic condition. We have to provide the photoperiodical as well as some air conditioned provisions also we have to provide. The light intensity should be 200 lux to 1600 lux for the plant growth for the photosynthetic purposes. And instruments room, we know that we have basic instruments like stirrer, orbital shaker, then bottle cleaner, and pH meter, and thermometer, weighing balance, and spectrophotometer, calorimeter. Some of the basic instruments we have to maintain in the room for the some analytical purpose also. And glasswares and other requirements for the plant tissue culture, the glass bottles, petri dish, purette, funnels, beakers, and pipettes, conical flasks with screw caps, graduated measuring cylinders are provided, spatula, scalpel, and forceps, and wrapping papers, buckets, gloves also we have to provide. And the practical manual of the plant tissue culture should be available in plant tissue culture laboratory. And in very common and as well as Indian popular plant tissue culture manual, the plant tissue culture theory and practice revised edition by the S.S. Bouchwani as well as M.K. Razdan, and plant tissue culture manual by the Lidsey, and others methods in plant tissue culture by the Kumar. These some examples manuals have to be provided in the plant tissue culture laboratory. Suppose if you are going to start a laboratory that a low cost effect, then what is the economics of this plant tissue culture laboratory we have listed out. General chemical cost is glasswares we need 15,000, all chemicals we need 25,000, aluminum racks we need 10,000, other requirements for the laboratory we should have at least 25,000, and instruments we need a water purifier system about 15,000, electronic balance we need 3,000, and pH meter we need 3,000, hot plates 1,000 rupees, and refrigerator we need at least 15,000, laminar airflow chamber we need 25,000, pipette micro pipettes we need different ranges that we need 10,000 rupees, for autoclave we need 20,000, and computer we need 20,000 for the starting of a low cost plant tissue culture laboratory. And also other facilities we need to run the plant tissue culture laboratory with the hand washing facilities in outside the lab, and fire extinguisher with a system. The waste disposal area should be provided with a different mode. Thank you very much for patient listening. Thank you.